Okay, we are now on part 2 of aerobic metabolism. This is going to continue with talking about ATP production, but now we're solely focused on the electron transport chain. So let's go ahead and get started. Okay, we're going to talk about the electron transport chain, and this is where oxidative phosphorylation comes in. And then we will continue with some other means of ATP production with beta oxidation and protein oxidation, and then Cori cycle and lactate shuttle. All right, so before we kind of go over all of what's going on here, oxidative phosphorylation occurs in the mitochondria of the cells, and it uses their structures, enzymes, and the energy released by oxidation of nutrients, which we said was turning one substrate into something else and releasing electrons. And that helps to produce more ATP. 90% of the ATP produced in energy metabolism takes place in the electron transport chain. So this picture here shows that the electron transport chain takes place in this inner membrane here. So it's going to take place inside the mitochondria. And that's what this picture here shows. If you see, this says outer mitochondrial membrane, this top part here. So the outer membrane, and then here shows the inner membrane down here. So we're basically in between the outer and the inner membrane. So that's where this takes place. And what this process is, it's actually the pumping of hydrogens and the passing of electrons through the inner and outer. So it's passing it back and forth through these different pumps. And these pumps exist in that inner membrane space. So electrons captured from NADH and FADH, which we learned about in glycolysis and in the Krebs cycle. So those are taken, so those structures are taken and transferred through these complexes. So on the next slide, I'm going to go over all of what we see kind of going on here, but here I'll kind of talk about like the labeling of each of this. Actually, I'll just kind of go over everything here. So we already know we're going to be working with the outer and inner membrane space, which I just described, and we'll talk about the pumping of those hydrogens. So the electrons carried by NADH enter the electron transport chain first, prior to this first pump. They're going to produce more ATP than the FADH, where the FADH are going to enter in the second pump, so after the first. So electrons and the hydrogens are passed from the beginning to the end of the electron transport chain down a concentration gradient from higher to lower concentrations of hydrogen. This is going to release energy as they pass from one cytochrome to the next down the electrochemical gradient. So let me kind of just start here. Okay, so these NADHs, we know we've already been, the Krebs cycle was in the electron transport chain, was in the mitochondria. So we're already in this structure here. So the NADHs then just enter into the next part of the mitochondria, and the NADHs are going to drop off their hydrogen. And so we see that here. We see that NADH is then going to turn back into NAD. It's going to lose its hydrogens and its electrons. So hydrogens have associated electrons with them, and they're both used in this process. So down here, there's a low concentration of hydrogen, and up here, there's a high concentration. So one thing we kind of want to understand is that the hydrogens, once they're dropped off, they're pumped through, or kind of they're passed through this first pump. And as they're passed through, we see an electron there. It kind of stops here in the middle of the pump, and it doesn't get passed all the way to the outer membrane. It stops in the middle, and it's passed along the inner membrane. So that's when the NADHs, so all the NADHs kind of drop off their hydrogens here. So of these three pumps, in the inner membrane, for every 2 electrons that move along the ETC, each of the 3 pumps move electrons from the inner to the outer compartments. So the first and the second pump, which we see first pump, second pump, those are going to pump 4 hydrogens through each one of those. And the third pump we see here will pump 2 hydrogens through. So all those hydrogens, remember, came from the FAD and the NADHs. So they pump those hydrogens through, resulting in a high concentration in the outer membrane because there's a lot of hydrogens now. And then the electrons are passed through this here. So they don't get pumped to all the way through the top. So the hydrogens are pumped to the top and the electrons are kind of passed through this middle part. So first structure we see is a ubiquinone. A ubiquinone is a mobile transfer molecule of electrons. So it's just a transfer structure here. So the electrons are passed to the ubiquinone, in which then are passed through the next pump. And then the electrons then are passed to a cytochrome. So the first is cytochrome C. A cytochrome is an iron-containing hemoprotein. The heme group is primarily responsible for generating ATP. So the electrons are then kind of passed again to that next pump there. So this whole process creates a potential energy kind of gradient used to phosphorylate ADP at the very end of the system. So the ATP is produced, but only if ions move down the gradient from the inner to the outer compartments. So this is our gradient here. We're passed from one to the next. So from this high concentration of hydrogens at the top, they're then going to pass down through this respiratory assembly. So the respiratory assembly will pump the hydrogens back through, and when that happens, they're going to activate an enzyme called ATP synthase. So this ATP synthase will then take this ADP, and it's going to become phosphorylated. That's where the oxidative phosphorylation occurs, and that produces the ATP. We see over here, oxygen is going to act as the final electron acceptor. So we see there's our oxygen, there's our 2 hydrogens, and when those electrons, we see this electron here pass to the ubiquinone, to the cytochrome C, back down to the bottom of this third pump. So oxygen will accept those final, is the final electron acceptor. So the 2 hydrogens plus the half oxygen will form our H2O, which we see water is formed at the very end. Water is the kind of end product here, and this is needed to keep the electron chain functioning. All right, so continuing. The pumping generates that gradient that I talked about, and that gradient's used by the ATP synthase complex, which is our respiratory assembly right here, and this allows for the ATP production. So as hydrogens pass through the respiratory assembly, we see they're pumped back down. They activate ATPase, or ATP synthase enzyme, which phosphorylates the ADP to make more ATP. So we're going to watch this quick video about mitochondria and kind of all this, but then we'll watch a better video later.
In the mitochondrion, the energy stored in NADH is used to generate a proton gradient across the mitochondrial membrane, and the energy of the proton gradient is used to make ATP. When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD and FAD are reduced to NADH and FADH2. Inside the mitochondrial matrix, the electrons from NADH are transferred to the electron carrier coenzyme Q by NADH dehydrogenase, and the protons are transferred across the membrane to the intermembrane space. Coenzyme Q carries the electrons to the cytochrome BC1 complex. As the electrons move from the BC1 to cytochrome C, more protons are carried from the inside to the outside of the membrane. Electrons are also transferred from FADH2 to coenzyme Q, with the protons being transferred across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen, the terminal electron acceptor, and water is formed as the product. The transfer of protons to the intermembrane space generates a proton motive force across the inner membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
Okay, so I hope that made a little bit more sense. I have one more picture here that some students like better because it makes a little bit more sense. So I'm going to explain this one last time and then we'll watch a final video over all these systems. So this basically here, we look at one. So complexes of ETC function to pump hydrogens from the mitochondrial matrix to the inner membrane space, and we see that here. Matrix to that inner membrane space, and then remember up here is the outer membrane, down here is the inner membrane. So the NADHs come and they drop off the hydrogen, which then results in the NAD, and then the hydrogen you see here is pumped to the top. So here's our proton pump. FADH drops off, it can't go into the first pump, but it can go into the second pump. So that's FADH is then pumped up through there. 2, the hydrogen gradient is formed with a higher concentration of ions in the inner membrane space, which is up here, compared to the matrix, which has a lower concentration. And then you also notice here this concentration gradient where we have our electron carriers, and then they pass the electrons all the way down through that last to that last pump there. And then we already mentioned that water acts as the final electron acceptor. So we see the electrons are passed through here, and then, oxygen acts as the final electron acceptor. So we have our 4 hydrogens, we have our electrons that were passed back through, we have oxygen, which will then form H2O. We said H2O is the waste product that helps keep the cycle regenerating. And then last over here, the hydrogens will flow back down into their matrix, or back down through the matrix through the concentration gradient, which will activate the ATP synthase enzyme, and that will cause the phosphorylation of ADP. So we have phosphate, which is added to adenosine diphosphate to make adenosine triphosphate. And so this process keeps happening over and over again, resulting in a high capacity of ATP. Okay, so overview of ATP production. So glycolysis, we already know what we produced here and in the intermediate step and in the Krebs cycle. We went over that in the last video. So thus far, we already know how much we have before we start the electron transport chain. So what we need to know though, what I haven't gone over, is that one NADH, when it drops off its hydrogen and associated electrons, that's going to actually equal 2.5 ATP. So we have 10 NADHs, which we see right there, times 2.5 equals a total of 25 ATP. One FAD equals 1.5 ATP, and we see that we had 2 of them. So 2 FADs times 1.5 equals 3 ATPs. So we have 28 ATPs from the electron transport chain alone. But we have to add the 4 to 5 ATP that we got from those other steps. So when we add that together, we get a total of 32 ATP if we use glucose and 33 ATP if we use glycogen for that total production. So one molecule of each of those will equal that much ATP. The one thing to know is every book actually gives a different number. So this is from the exercise physiology book that was required for this class. This is the number you get. But the book I used to teach advanced strength training, the 4305 class, formerly 3322, that book actually gives a different number. I think it gives like 36 total ATP. So I want you to know it's a little different depending on the book that you read or the class or professor that you take. But for this class, those are the numbers that we use. So also one more thing I want you to know is this is from one molecule of glucose. Think about how many glucose molecules we consume in a given day, like billions to trillions of glucose molecules that we consume. So think about it in that concept. This is just from one glucose molecule. Next we see here is, if you like charts, I'm not a big chart person. I kind of like what we just did. But if you like looking at charts to figure out ATP production, this is a great chart to learn. So it tells you ATP lost if we use glucose or glycogen, and we see 2 if we use glucose, 1 if we use glycogen. And then we see how much ATP was produced in glycolysis. With each source, we get 5. And then how much ATP from the NADH molecules, 5. And then we see here, pyruvate to acetyl-CoA, we got 2 more NADHs, which equals a total of 5, based on the 2.5 ATP per 1 NADH. And then we see here the Krebs cycle, how many we produced, the NADHs, giving us a total of 32 or 33. And it assumes that 2.5 ATP per NADH and 1.5 per FADH, which we already learned. But yeah, so that just shows you the calculation again. So again, I do like charts. So this here is basically to have you draw out the electron transport chain however you think learning it best would be. Just knowing total production of ATP, NADH, and FADH and how the system actually works. Again, you don't have to do this, but I think drawing is a really great way to learn. So here we're going to do kind of a neat critical thinking question. So we're going to watch this video, and it says, based on your current understanding of what we just learned about aerobic metabolism, discuss how the theory behind this new supplement, CoQ10, may provide health benefits or increase exercise performance. So I actually take CoQ10, and this video here explains why it's actually really important.
Mitochondria are little power stations inside your cells, working hard to produce the energy you need every day. But there's a byproduct of their energy production, free radicals, and while they might be free, they can be costly. They're rogue molecules generated within the mitochondria, and they can damage the delicate equipment inside your cells. The good news is mitochondria are clever, creating internal barriers of molecules that neutralize free radicals. But as we age, mitochondria can't reline their barriers fast enough to cope, and some free radicals can escape. This can cause mitochondrial dysfunction, and this can have some serious effects. It is known to be a significant cause of a large number of health conditions, including 7 of the most costly to human life. In fact, the cumulative damage caused by malfunctioning mitochondria is one of the primary causes of aging itself. Nothing has been able to solve this problem at its source, until now. MitoQ has made a significant scientific breakthrough, and it's a world first. Here's how it works. It all begins with CoQ10, a versatile molecule mitochondria use in many ways, including in their defensive barriers, which, as we've seen, can be tricky to maintain. So what if mitochondria could just call for help? And a bunch of CoQ10 repair molecules came around. That'd work, right? It would if it wasn't for a 2-layered problem, the double membrane, mitochondria's top-notch security system. These two keep a strict eye on what is and what isn't allowed to come inside the mitochondria itself. They have a simple checklist. To enter, you have to be small, And you have to be useful. Unfortunately, when outside of the mitochondria, CoQ10 doesn't check either box. Even if they bother to get fit, the double membrane doesn't forget a face. So what now? Well, luckily there's a super slick ion called lipophilic triphenylphosphonium. Phil's positively charged, all charisma, and a great motivator for CoQ10. Because once they get in shape, Phil gets them looking irresistible. And they'll bond together, forming an epic crew. When this crew rocks up to the double membrane, they pass the security checklist easily and are welcomed in. Slim CoQ10, bonded with Phil, is a dream team, known as mitoquinol, or MitoQ for short. They're our breakthrough, the key to improving mitochondrial health. Without fitness or Phil, CoQ10 aren't much help at all. But as MitoQ, they can penetrate mitochondria hundreds of times more effectively, getting them the support they need right where they need it, helping to realign the defensive barriers, which minimizes free radical leakage and damage, and decreases the risk of health issues associated with mitochondrial dysfunction. When mitochondria are under less stress, energy production returns to optimum levels. Your cells become revitalized and rejuvenated, so you can live your best life possible. MitoQ powers your cells.
All right, so pretty interesting. Normally, if we were in class, we would get into groups and y'all would discuss this and we'd talk about it and kind of all that. But just something for you to think about. Okay, next we want to talk about a system called the Cori cycle and then another one called the lactate shuttle. So first, with the Cori cycle, also called the lactic acid cycle, is basically when lactate is produced by skeletal muscle and other tissues, it is transported to the liver to make more glucose. And then that glucose can be used to maintain blood sugar or for liver glycogen. So lactate is transported via the bloodstream to the liver to make more glucose, utilized to maintain our blood glucose levels, or if we actually need more stored glycogen. So that's what this kind of process shows right here. So let's take the muscle and fast glycolysis. So we know glucose or glycogen converted to glucose and it's put through our glycolytic system and it produces pyruvate. But during fast glycolysis, the pyruvate accepts the hydrogen and it becomes lactate. Well, lactate's going to build up in the bloodstream, making it really acidic and making it harder for us to exercise. But a way to clear lactate out of our system is to send it to the liver. And the liver can actually use lactate to either make more glucose for our blood glucose levels, and it can be sent back to the muscle to make more ATP, or it can be stored as glycogen so we can use it for fuel later. This here also shows the Cori cycle, so the blue arrow Cori cycle. So from high intensity exercise, we have lactate, which can actually be converted back into pyruvate, which can be converted into glucose, which can be stored in the liver as glycogen, or it can be sent back in the blood, back to the muscle for exercise or rest for energy. So what's the purpose of this? Purpose is to prevent metabolic acidosis in the muscles and then help produce more ATP. Metabolic acidosis is just when too much acid accumulates in the body due to extremely high intensity exercise. But this helps us to clear away the acidity. So it actually makes lactate more of another way to just be used in the body rather than an acidic byproduct. So that's the first kind of process, is the Cori cycle. The second one is very similar, and this is the lactate shuttle hypothesis. So this is a kind of a theory, because it's a hypothesis. It's basically once lactate leaves the muscle and enters the bloodstream, it can not only be utilized by the liver via the Cori cycle, but it can also be used for other organs, including the heart and the brain. So lactate essentially becomes a precursor for oxidation. Our brain uses about 25%, or is made of about 25% of glucose, which it needs that to function. Glucose is the preferred fuel source for the brain. So we can take lactate and make more glucose, possibly for the brain and other organs. So now lactate is viewed not as a waste product, but a way to shuttle carbohydrates around the body and be used by other tissues. So yeah, let's look at this process here. So we already talked about the blue arrows, which is the Cori cycle. So now let's look at high intensity exercise and the lactate shuttle. So we know that high intensity exercise causes glucose to turn into pyruvate, which can create lactate, where lactate can travel in the blood. And when lactate's high in the blood, we want to clear it, because that's going to create acidity, just like we talked about. But now lactate can move intercellular, so within cells, and then intracellular versus between cells, intercellular. So we can send lactate now back into pyruvate, so where inactive muscle, heart, kidneys, brain can use it for fuel sources. Again, the purpose of this is like the same as for the Cori cycle. It prevents a buildup of lactate. So it just gives extra fuel for working muscle in the form of glucose. So you probably get it at this point. We can take lactate and turn it into pyruvate. And then we already know pyruvate can turn into lactate, pyruvate can turn into glucose, glucose into pyruvate. So kind of everything can turn into each other, because we know oxidation occurs, and oxidation can occur to any of these these fuel sources. Oxidation, we know, is just undergoing a reaction where electrons are lost to form another product. And so that's what's happening. All these are being oxidized to form something else. So yeah, that's the lactate shuttle and the Cori cycle. So let's move on to some other kind of topics involving the Krebs cycle that you may have noticed when I went over the Krebs cycle, and that's protein oxidation and beta oxidation. You probably saw this right here, and I didn't talk about it because I wanted to save it for this slide. Okay, so beta oxidation we see right here is when fatty acids are broken down and transformed into acetyl-CoA. Because when we just learned about the Krebs cycle in the last video, we talked about how glucose, we know from glycolysis, turned into pyruvate at the end of glycolysis, and then the intermediate step we produced acetyl-CoA, and we went through the Krebs cycle. So we basically only talked about how glucose kind of goes through the Krebs cycle. Beta oxidation explains how fatty acids go through the Krebs cycle. So they have to be transformed into acetyl-CoA just like pyruvate did. And then that process here we'll discuss on the next slide. We'll go over it in detail. But we see here that a FAD is produced, and so is an NADH to get that acetyl-CoA. Protein oxidation, you probably also noticed, is right here where we have our amino acids. So this is where we can turn amino acids into different sources. So we can turn them into pyruvate, which can go through that same process, or they can enter into the cycle directly as oxaloacetate, fumarate, succinyl-CoA, or alpha-ketoglutarate. You'll probably learn more about this process in your sports nutrition track class, where you actually talk about all the different types of amino acids and how they can become oxidized, losing their electrons and turning into another product like the kind of products that you see here in the Krebs cycle. So this class doesn't go over the amino acids specifically. It just explains how amino acids can enter the Krebs cycle. And then later when you take that sports nutrition class in the kinesiology department, you'll learn more about what actually has to take place to the amino acids for them to undergo that process. But I'll go over it a little bit more in detail. This is just kind of the introduction slide to show that there are other processes that kind of occur here. So let's first talk about beta oxidation. So beta oxidation, we first have to start with this enzyme called hormone-sensitive lipase. So we learned back in chapter 2 that triglycerides are a type of lipid, which is just a type of fat, inside our fat cells, our adipose cells. And they have to be broken down into glycerol and free fatty acids. So we know that 1 glycerol and 3 fatty acids equals a triglyceride. The enzyme hormone-sensitive lipase will break the triglycerides into these 2 parts here. We learned in chapter 2 that glycerol cannot be used by the muscle to produce ATP, but it can be sent to the liver for the Cori cycle to make more glucose, and then that can be come back and used to make energy. But free fatty acids, that can be used by the muscle, and that can enter into the beta oxidation process, which we'll go over on the next slide. So when hormone-sensitive lipase breaks apart triglycerides, the glycerol and the free fatty acids are released into the blood. So other cells can take up the lipids from the blood to use for aerobic metabolism. So free fatty acids enter the muscle fibers by diffusion. Diffusion is the net movement of anything from a region of higher concentration to a region of lower concentration. Diffusion is usually driven by a concentration gradient. The concept of diffusion is widely used in physics, chemistry, biology, kind of everything. So here, this is kind of like chemistry, what we're looking at. So free fatty acids can just diffuse into the muscle fibers directly. Let me go over this chart, or this kind of picture right here. So we've probably already know the word lipolysis, lysis meaning breakdown, lipo meaning lipid. So this is the hydrolysis, or the breakdown of lipids. So this here is in the adipose site, which is a fat cell, or adipose tissue. So this is fat. We have our triglyceride molecule. So we see here hormone-sensitive lipase is going to break apart the triglyceride, and then we have a free fatty acid and we have a glycerol molecule. Hormone-sensitive lipase is manufactured by the pancreas, and it acts to hydrolyze different types of free fatty acids that are found in oils and in fats that we consume. Hormone-sensitive lipase is sensitive to functioning or fluctuating hormones such as insulin and epinephrine. So if you see here, we see epinephrine, which is in the bloodstream. We see this process here, cyclic AMP and protein kinase. We'll talk about that not until chapter 8, where we get into hormones with epinephrine and kind of all this process here. But what you just need to know is hormone-sensitive lipase breaks apart this here. The purpose is to mobilize stored triglycerides under high energy demand and release the free fatty acids into the blood to be used in aerobic metabolism. Fats also generate a higher amount of energy due to the large storage of adipose tissue in the body. The body stores a lot of fat. We talked about that a little bit in chapter 2. So there's a lot of fat stores in the body that we can use for energy during aerobic metabolism, so at rest or low intensity exercise. So here's the beta oxidation process. So we see fatty acids and glycerol can both be metabolized here, but just differently. So I already mentioned that glycerol is transformed by the liver into glucose, which can be used to form pyruvate, and then it can be turned into acetyl-CoA. But the free fatty acids can't enter directly, and that's what this picture shows here. So fatty acids broken down into acetyl-CoA. This actually requires 2 ATP to start the process. So for per fatty acid, just like we lost 2 ATP with glucose, same thing with free fatty acids. So we still lose those 2 in the beginning. And then once we start the system with the 2 ATP, we can generate 1 NADH and 1 FADH during that process. Again, just like glucose did, it produced 2 NADHs per glucose molecule. Here we produce an NADH and an FADH. So we see here we've turned it into our fatty acetyl-CoA. And it's going to go through all these different steps in the mitochondria. And then by the end of it, so once we put it through the Krebs cycle electron transport chain, we get ATP. But the amount of ATP produced depends on the specific fat that was oxidized. We already mentioned that lipid produces substantially more ATP than glucose does. So more ATP than our glucose metabolism. It's been kind of shown that one fatty acid can produce up to 130 ATP per 1 molecule, whereas with 1 molecule of glucose, we said produces about 32 to 33. Well, one fatty acid can produce about 130 ATP. We're not going to actually learn that process here. It's just kind of important to know that kind of the beta oxidation process of how we can use free fatty acids instead of glucose. All right, so this next slide here shows a picture of how we can produce lipids from other sources, so like glucose and amino acids. And normally I would turn this over to the class and basically ask this question: can you gain fat mass simply from ingesting protein and carbs only? And you're supposed to explain why. So normally I would let you guys discuss this for about 5 minutes and come up with an answer. So maybe pause the video and look at this chart right here and think about, is this possible? So if I only eat protein and carbs, can I gain fat tissue? And by looking at this, the answer is yes. So let's talk about how. So first, kind of let's look at our glucose. Glucose can be transformed into glycerol via oxidation, and then that can be turned into acetyl-CoA to make triglycerides via the adipose tissue. So that is actually the synthesis of fatty acids. Amino acids we see down here can be used to make pyruvate and can become glucose, and then glycerol or acetyl-CoA and can also be utilized to synthesize or make more fatty acids. So the answer is yes here, and we can see following all these arrows, and I mentioned it earlier, that one substrate can turn into another product very easily via oxidation, the loss of electrons and hydrogens, and the arrangement of its atoms. So because of the ability to utilize both carbs and proteins to make free fatty acids and glycerol, and eventually form triglycerides, it's possible to gain fat mass even when just ingesting carbs and protein, because the body basically can turn one substrate easily into something else. If the body doesn't use our glucose that we consume, it's going to be stored as glycogen. When our glycogen stores are full, the glucose we consume is then turned into triglycerides, which are stored as fat if our glycogen stores have surpassed. If the body doesn't use protein to repair our broken down muscle or to build our muscle, then the body will store that extra amino acids, that extra protein, also as fat. So fat storage comes from when we consume too much protein and carbs or fat that the body doesn't actually use. So if I'm supposed to eat X amount of calories a day and I consume more than that on a daily basis, then that extra energy that is in my body has to be stored somewhere, and it's going to be stored as fat. So that's kind of how that works, and you'll learn more about that in nutrition as well. This picture here goes into more detail about that process that I just mentioned. When we intake food, so fats, carbs, and proteins. Let's start with proteins. Proteins turn into amino acids, and then we can use amino acids for the body. So we have our body, our muscle protein, and then we can break down the protein in our muscles and that turns into an amino acid pool. We can also use that amino acid pool for protein synthesis to build up the body's proteins. So this process occurs, and then this amino acid pool we can use in metabolism, which we'll learn about in the next video. Actually, on the next few slides, we can learn about it, where we can use amino acids as energy. Also, we can take amino acids and they can go through a process called gluconeogenesis, which we learn on the next slide, which is turning a non-carb substance into glucose. So amino acids, gluconeo means it's a new source. So amino acids can turn into glucose. All right, let's look at carbs. So carbs, we know, are broken down into glucose. And if there's excess glucose in our body that's not used, that goes through lipogenesis, which is the formation of lipids, which go into our fat stores. We can also use glucose, which are stored as glycogen. So glycogen storage in our muscle and our liver. And then we also have a glucose pool, which is the circulating blood glucose in our body. So this process we've learned about in chapter 2, where we have glycogenesis, which is taking that glucose pool and forming our glycogen stores when the glycogen stores are low. Or let's say the glycogen stores are high and we need more blood glucose, we'll go through glycogenolysis. And glycogenolysis is the breakdown of glycogen to add to our blood glucose that's circulating in our body for energy. And then we can use that glucose here for metabolism. And then finishing with fats. So fats are broken down into free fatty acids and our glycerol. And again, we can have our fat stores or our fat pool. So the FFA pool is the circulating fatty acids in the body, which can be used for metabolism for energy. And then the fat stores, let's say our fat stores are really high and we need to use the fat for fuel. We go through lipolysis to break down the fat, and then we can use the fat or the free fatty acids in metabolism. Or if there's a high amount of fatty acids in the bloodstream, they're going to go through lipogenesis, which creates our fat stores when there's too much fat circulating in the bloodstream. So I know I just went over a lot in this picture, but it's a really, really neat process of how we use all these different macros for metabolism. So if you need to rewind this and go over this again, it's an extremely important concept and topic to understand in detail to finish up this chapter. Okay, we're going to kind of finish up with metabolism of protein. And then that'll be the end of part 2. Okay, so I already mentioned gluconeogenesis or gluconeogenic is when amino acids are transformed into glucose. So that neo means it's a non-carb source or like a new source. The amino acids are also converted into pyruvate and acetyl-CoA, which we saw in that protein oxidation slide a little bit ago. So kind of let's go over this process in detail. So protein can provide energy through several different pathways, but before an amino acid enters into a bioenergetic process, it must first have its amino group removed via transamination or deamination. And I'll go over what each of those mean in a second. But first, in order to assess the rate of protein metabolism, the amount of nitrogen excretion must be determined. So when we consume too much protein over what the recommended intake for the body is, then the body will excrete that excess protein as nitrogen. Because I mentioned in chapter 2 that amino acids contain nitrogen, which the other macronutrients don't. And so when we consume excess amount of protein, we can determine how much protein the body has used based on how much nitrogen is excreted in our urine. And so that's a test that's done in some research studies that kind of look at protein breakdown in the body. Okay, so transamination is when the amino group is transferred to a keto acid. So we know that with an amino acid, there's an amino group and there's an acid group. So transamination turns the amino into a keto acid. A keto acid is an organic compound containing a carboxylic acid group and a ketone group. So most all amino acids can undergo this process. The process determined by amino acids being used to make glucose. So that's kind of the purpose of this process here. The second is deamination, which is this picture that's shown. So this is when we take the amino group and instead of turning it into a keto acid, it creates ammonia. So this is the removal of an amine group from a molecule, breakdown of amino acid if there's excess protein in the body, like I mentioned. This occurs in the liver and the kidneys, depending on which amino acid is used. So we see here, it takes that amino group away, and then it creates ammonia. So ammonia is a base in the body, and it can upset the acid-base balance. To prevent this disruption of the acid-base balance, the liver combines 2 ammonias with 2 carbon dioxides like you see here, and that will create water and urea. So urea is in the bloodstream, and then it's excreted in the urine through the kidneys. So that's the process of each of these, and that is protein metabolism. So that's how we can metabolize protein. This is not something we want to do, but obviously it can be done. So amino acid metabolism is small. Like I said, we don't really want our amino acids to be metabolized because that may cause a breakdown in our muscle or a prevention of muscle building to occur. So when does amino acid metabolism occur? Extreme dieting. So extreme dieting meaning if we ingest fewer calories than needed to support the body's functions, then amino acids may be taken from our tissue and from our skeletal muscle for energy, and they can make glucose. This does result in muscle loss with extreme dieting. So when we go through really extreme diets, it's suggested to increase your protein intake so this doesn't happen. We don't want to lose our muscle tissue, but if there's not enough fat and carbs that are ingested, then we turn to what's available, and that's going to be the muscle, which is made of amino acids. Next is amino acids are metabolized when we have high protein diets. So this has to do with availability. So this increases amino acid metabolism due to the high availability compared to carbs or fat. If eating like a low carb diet and a high protein diet, there's not going to be very much carb or very much fat, but there's a lot of protein because that's the only thing you're ingesting. And so it's most available and it's easiest for the body to grab. So high protein diets, the body's going to take that protein that you ingested and use it for fuel because it's available. And then last is long-term endurance activity. So long-term endurance activity will activate an enzyme called proteases. This enzyme is capable of degrading protein found in skeletal muscle. A small increase in amino acid metabolism will occur if low blood glucose or glycogen stores, or if there's low blood glucose levels or low glycogen stores, then the body has to turn to something, so it's going to turn to protein or amino acids. But just know this percent is small. For all of this, it may be up to about 15% of metabolism will come from protein, especially during the activity. All right, so here's a study right here. And normally I would just show this study to the class and then have y'all talk and then ask what conclusions can be drawn from these findings. So what I would do if I were you is pause this video and then kind of read through this study here. And it's kind of hard to understand because I've kind of like abbreviated everything. But then kind of just figure out what conclusions can you draw from this data. So once you pause the video and go over this for a little bit, I'll answer the question. Okay, so 12-week study. It was a study was based on men, and they had 3 different groups, and on average we're looking at how much weight all the men lost, and it was about 19 to 21 pounds, but it's different based on the group that you were in. So this here, it says a diet group. So this says that the group of men, they only dieted. They lost 69% of their weight that was lost was from fat tissue. And then the diet aerobic exercise group lost 78% of weight from fat. And the diet aerobic and resistance training group lost 97% of their weight from fat tissue. So what does this mean? So this is saying like out of the 100% of weight loss, 69 was fat, meaning that that other percentage was from muscle, our muscle tissue or our glycogen stores, but mainly referring to muscle tissue. So if you diet only, you're only going to lose about 70% of there's 70% of the weight you lose is from fat, whereas about 30% of the weight you lose is from your muscle tissue, which you don't want to do. If you only diet and do aerobic exercise, that does increase now, so we're burning more fat, but there's still about 20% of the weight you lose is also going to be from your muscle. This part here is the most important. This just says that if you resistance train, RT is resistance train. So if you do some dieting, aerobic exercise, and resistance training, the weight you lose, 97% of that is fat, meaning only 3% of the weight you lost was from your muscle tissue, because the resistance training helped to maintain your muscle while you were dieting and trying to lose weight. So that's why it's important to resistance train and lift weights when you're trying to lose weight, because that helps you maintain your muscle. If you don't add that resistance training component, some of the weight you lose is going to be from your muscle tissue, which we see 20 to 30% of it was for these other two groups. So that's really kind of all this showed right here. But yeah, that's the end of part 2. I know this was a little bit longer video, and let's move on to part 3.